Lipoproteins, introduction, classification and function. You are watching this lecture in my YouTube channel, Dr. Biochem Lecture. My name is Dr. Alamzev. I am a professor at the Department of Biochemistry, University of Malacca. The contents of today's lecture include introduction to lipoproteins, structure of lipoproteins, classification of lipoprotein, and examples with functions. First, introduction. Lipoproteins are biochemical assembly made of lipids and proteins. Generally, lipids are insoluble in plasma. In order to be transported, they are combined with specific proteins to form lipoproteins. These are complex molecules having complex structure. Many enzymes, transporters, structural proteins, antigen, adhesion, and toxin are lipoproteins in nature. Structure of lipoprotein. A lipoprotein is made of two main components. One is known as lipoprotein core, which is made of triglycerides and cholesterol or cholesterol ester. The second part is lipoprotein surface, which is made of phospholipids, proteins, and cholesterols. The yellow color is the inner hydrophobic core of the esterified cholesterol. The protein present in the lipoproteins are called lipoproteins, which are weakly associated with a particular lipoproteins and are easily transferred to another lipoproteins of a different class. In this table, you can see different classes of lipoproteins. Lipoprotein A, lipoprotein A1, A4, B48, ApoB100, ApoC1, C2, C3, ApoD, and ApoE, and having different molecular weights. The high molecular weight, the highest one is ApoB100, followed by ApoB48, and their presence in different lipoproteins. Now, classifications of lipoproteins. There are 5 classes of lipoproteins. All these classes contain characteristic amount of triacylglycerol, cholesterol, cholesterol esters, phospholipids, and apoproteins. These 5 classes are based on their density, and the density is based on the composition of the amount of this component. Most proteins have density higher than 1.3 to 1.4, and the lipid aggregate have usually density of about 0.8 gram per milliliter, which is lower than that of the proteins. The first class of the lipoprotein is known as chylomicron. They are also known as ultra low density lipoprotein, ULDL. They consist of triglycerides 85 to 92%, phospholipid from 6 to 12%, cholesterol from 1 to 3%, and protein about 1 to 2%. Chylomicron has a diameter. They are big in size, 75 to 1200 nanometer, and density is less than 0.96 gram per milliliter. They are assembled in intestinal mucosal cells. They enter lymphatic system and enter the blood through thoracic duct. Structure and composition of chylomicron. In this figure, you can see the white surface area is made of phospholipids, while the yellow center or the triacylglycerols, so they contain high amount of triacylglycerol, while the blue embedded or the apoproteins present in the chylomicron molecule. Functions of the important function of chylomicrons are that they are found in the endoplasmic reticulum, in the enterocytes of the small intestine, from where they do their function. Apoprotein specific to chylomicron is ApoB48. Chylomicron basically transport lipids absorbed from the intestine to adipose, cardiac, skeletal, and other tissues, and these lipids are usually dietary lipids. You can see here from the intestine they take the dietary lipids and then take it to the lung, from where they reach the blood, and the lipids are transported to the adipose tissues, where they are converted to different for the metabolic purposes or for the sake of energy, and then the chylomicron is reused. Second class of lipoprotein is known as very low density lipoprotein. They are synthesized in the liver. They contain mostly triacylglycerol, but with a significant amount of cholesterol and cholesterol esters. They have a diameter of 30 to 80 nanometer and density less than or about 1. They convert it to LDL, which contain an increased proportion of cholesterol and cholesterol ester. Structure of VLDL. In this structure, you can see the center, the core contain the green and yellow atoms, which represent the cholesterol ester and triacylglycerols, and the apoprotein C, apoprotein E, and apoprotein B 100 molecules, and the surface on the surface in the phospholipids and function of VLDL. They transport lipids from the liver to the tissues, and especially those lipids which are endogenous in nature, like triglyceride, phospholipids, cholesterol, and cholesterol esters. Cholesterols in the LDLs referred to as a bad cholesterol, since LDL are implicated in atherosclerosis. That's why it is important for health. The third class is known as intermediate density lipoprotein, or IDL. They contain protein about 18 to 18%, phospholipid about 22%, and triacylglycerols about 31%. IDL is secreted by the liver. They contain several apoprotein E and a single copy of ApoB100. The multiple copies of the apoprotein E allow IDL to bind to LDL receptor with a very high affinity. IDL is usually 20 to 35 nanometer in diameter and a density above 1. They are metabolized by either endocytosis or by lipase. Function of IDL. IDL basically transport triglycerides to adipose tissue and muscle. They are converted to LDL during circulation. You can see here in this figure, and the VLDL upon the action of lipase form IDL, which is converted to LDL, and then it is transported to the extrahepatic tissues, and from there it is converted to HDL, and then Alkate convert it back to IDL, which is back to the liver. The fourth class is low density lipoprotein, or LDL. This is very well-known type of the lipoprotein. It is made of about 25% protein, 21% phospholipids, and about 4% triacylglycerol. They can transport 3 to 600 fat molecule per particle. They contain ApoB100 and other proteins. LDL Particles are approximately 22 to 27.5 nanometer in diameter and have a mass of about 3 million daltons. Now the structure of LDL. In this figure you can see the atherosclerosis plaque, which is shown in the yellow, and the picture contain the blue is the apoprotein B100. Function of LDL. LDL delivers fat molecules to cells. LDL involved in atherosclerosis, a process in which it is oxidized within the walls of arteries, resulted in the heart disease. The fifth class is high density lipoprotein, HDL. This class is made of proteins about 33%, a high amount, phospholipid about 29%, and triglyceride 8%. They are typically composed of 80 to 100 proteins per particles. The lipid carried includes cholesterol, phospholipids, and triacylglycerols. HDL is the smallest in size, ranging from 5 to 17. They have a density higher than 1. They are the most abundant apoproteins, contain ApoB, A1 and A2. You can see the structure of apoprotein HDL with the apoprotein E, apoprotein A1, ApoA2, ApoC1, C2, C3, and the precholesterol, the cholesterol esters, and the triglycerol in the core. Function of HDL. HDL carries used cholesterol from the tissues, extrahepatic tissues, back to the liver. They also donate some cholesterol ester to circulating VLDL for redistribution to tissues. HDL taken up by the liver and degraded. The cholesterol is excreted as a bile salt or repacked in VLDL for distribution to tissue. Cholesterol synthesis in the liver is regulated by the cholesterol arriving through HDL, that is, and dietary cholesterol returned by chylomicron. Cholesterol in the HDL is also referred to as good cholesterol. Serum lipid profile. In human, the ideal and healthy level at the fasting condition of the serum lipids, that is total cholesterol, is usually less than 200 mg per deciliter. Triglycerides are less than 150 mg per deciliter. LDL or less than 100 mg per deciliter. HDL are usually 50 or higher. So you need to maintain your serum lipid profile for the healthy life.